Hello everyone. In this video lecture, we will start with locomotion and movement. What are the different types of muscles? How does the muscle contraction takes place? What is the mechanism of muscle contraction and what are the different types of muscles? We will go through the slides one by one and let's see what is locomotion, what is the movement. Okay. Now, locomotion. Basically, locomotion, it is the movement of an individual from one place to another. What is movement? Movement can be of any body part. Okay, if suppose if we are going to displace our hand from one position to another position, that is called as the movement. Okay, now see here, there are two types of movements. One is the induced movement and the another is autonomic movement. Induced movement means when, for example, there are the non-living things and these non-living things are moved from one place to another. For example, if I pick a stone and I change the position of that stone, so that is the induced movement. I'm inducing the movement. And what are the autonomic movements? Autonomic movements are the movements by the body parts on their own. So the energy is produced in those organs and the organs are moved from one position to another position. That is the autonomic movement. Now, movement of body parts can be the movement of external body parts as well as the movement of the internal body parts. These are very common things. The movement of external body parts. Okay, for example, movement of the limbs, the appendages, head, trunk to maintain the equilibrium of the body. Okay, then in many animals, limbs and appendages also carry out locomotion. Movement of limbs, tongue, jaw, snout, appendages and tentacles are used by some of the animals. For example, tentacles in hydra are used by those hydras for capturing the food. The mouth is also used for capturing the food. Okay, then movement of the eyeballs, okay, help to see the object. However, movement of the pinna of the ear, they help to enable the animal to collect the sound vibrations. So these are some of the movements in the external body parts. What about movements in the internal body parts? Let's see some of the examples. Okay, movements of internal organs. Food and urine are moved through the digestive system as well as the urinary tract. Okay, and that movement is called as a peristaltic movement. Movement of the cardiac muscle enables the heart to receive and distribute the blood. Inspiration and expiration is done by the lungs. Okay, so there is also the internal movement. So these are some of the external movement of some of the external body parts and movements of some of the internal body parts. What are the types of movements? Types of movements are four types of movements are there. One is amoeboid, the second one is ciliary movement, muscular movement and the flagellar movement. What is amoeboid movement? The movement like the amoeba, okay, which is a protozoa. The amoeba moves with the help of the pseudopodia. So some of the cells in the human body, such as the white blood cells, the phagocytes, the macrophages, they show amoeboid movement. Ciliary movement, the ciliated epithelium which is present in the fallopian tubes of the female, it is present in the vasa efferentia in the male reproductive system, then the digestive system. So these ciliated epithelium, with the help of the cilia, they can transport the material from one place to the next place. For example, the ova, okay, that is a secondary oocyte in the female fallopian tube, is transported from one part of the fallopian tube to another part of the fallopian tube. As well as the sperms in the male reproductive system, they are also transported from one region to the next region with the help of the ciliated epithelium. The next one is the muscular movement. Muscular muscle cells undergo contraction and relaxation, so they help the body organs to move as well as also help in locomotion. What about the flagellar movement? If we see the structure of a sperm, the sperm has head, neck, middle piece and the tail. The tail shows whip-like movement, and with the help of the whip-like movement that is shown by the tail, the sperm can move from one place to another place. So that is a flagellar movement, and this we have completed the point of locomotion and movement. Okay, now what are the types of muscles? In the earlier topic also we have completed the point that is about the muscles. There are striated muscles which are also called as the skeletal muscles. There are smooth muscles which are also called as the visceral muscles, that is the internal muscles, and then there are the cardiac muscles. Okay, When we talk about the striated muscles or the muscle fibers, they are cylindrical in shape. Then they are syncytial in condition. Syncytial matlab there is more than one nucleus that is present. It is multinucleated condition. The nucleus may be present at the periphery. The skeletal muscle fibers, we have a diagram over here. See, you can see many nucleus are present in a single muscle fiber. Okay, then it also shows the presence of striations. Light bands and the dark bands are present. Light bands are made up of the actin protein, dark bands are made up of the myosin protein. Then these muscle fibers has plasma membrane. This plasma membrane is called as the sarcolemma. The plasma membrane is called as the sarcolemma, whereas the cytoplasm that is present, it is called as the sarcoplasm, and the endoplasmic reticulum is called as the sarcoplasmic reticulum. And endoplasmic reticulum in the skeletal muscle fibers, they store the calcium ions inside it. This calcium ion plays a very important role for the contraction and relaxation of these skeletal muscle fibers. Next one are the smooth muscles. Okay, one more point is that where the skeletal muscles are present. The skeletal muscles are basically present in the limbs, body wall, tongue, pharynx and beginning of the esophagus. Okay, now they are under the control of which nervous system? They are under the control of they are not under the control of autonomic nervous system. They are under the control of the animal's will. Okay, that is, the animal on its will can move the organs, and in all those organs the skeletal muscles are present. The skeletal muscles main what you can say is that the dominant mineral element is potassium. Okay, potassium is the dominant mineral element in the skeletal muscles. Along with potassium, calcium, magnesium, phosphorus is also present, and sodium is also present inside the skeletal muscles. After that, there is one more pigment that is present in the skeletal muscles, that is called as the myoglobin pigment. Myoglobin pigment, it is present in the muscle, that's why it is called as myoglobin pigment, and it binds with the oxygen molecules. Okay, so that is present inside it, and along with this there are also some energy producing compounds that are present. For example, ATP, then creatine phosphate are also present in the skeletal muscles. Next point is about the smooth muscles. Smooth muscles are also called as the visceral muscles, and they are not in those, they are present in those organs which are not under the control of our will, of the animal's will. For example, the digestive system, where the peristalsis takes place. Okay, then when we talk about all the internal organs, okay, for example, lungs, the urinogenital tract, urinary bladder, blood vessels, posterior part of the esophagus, stomach, then the dermis of skin, erector pili muscle of hair, and iris of eyes. Okay, these are some of the examples in which the smooth muscles are present. Smooth muscles, shape of the muscle fiber, it has a tapering end and only a single nucleus is present in its sarcoplasm. It also has longitudinal myofibrils that are arranged, and they are arranged parallel to one another. So myofibrils are present over here. So this is about the smooth muscles. Okay, and they are controlled by the autonomic nervous system, and hence they are not under the control of the animal's will. Next, about the cardiac muscle fibers. These are the cardiac muscle fibers. Cardiac muscle fiber shows the characteristics of both skeletal as well as the smooth muscles. Cardiac muscle fibers are uninucleated and they are cylindrical in nature, and these cardiac muscle fibers are connected to one another at some oblique projections. These sites, they have some dark disc shaped structure which is called as the intercalated disc. Okay, these intercalated discs, basically they play a very important role in multiplying or in sending the impulses from one muscle fiber to another muscle fiber. Okay, so मसल्स फाइबर्स के अंदर जो इम्पल्स का जो ट्रांसपोर्ट किया जाता है, दैट इज फास्ट बिकॉज ऑफ दिस इंटरकैलेटेड डिस्क, एंड एज दीज आर ब्रांच्ड एंड कनेक्टेड टू वन अनदर, द सिग्नल्स और द impulses can be transported very rapidly. So this is about the cardiac muscles, and these cardiac muscles also shows the presence of striations. They are present in the wall of the heart. Okay, so this is about the cardiac muscle fibers, and they also undergo contraction and relaxation. Skeletal muscle fibers undergo fatigue. Okay, but cardiac muscle fibers, they do not undergo the process of fatigue. Smooth muscles also do not undergo the process of fatigue. What is fatigue? Fatigue is a phenomenon when the skeletal muscles, okay, they're continuously under exertion. These skeletal muscles, in the absence or in the low concentration of oxygen, they start converting the glucose, they start utilizing the glucose. Glucose is converted into pyruvic acid, and finally the pyruvic acid is converted into lactic acid. Now when the lactic acid concentration increases inside the muscle cells functioning reduces, and that process is called as fatigue. Fatigue can be identified by pain and swelling of the muscles. Okay, so cardiac muscles and smooth muscles, they do not undergo the phenomenon of fatigue. Nextly, after this point, we are going to study the detailed structure of a skeletal muscle. Okay, now in the skeletal muscle, we have to remember that inside the muscle fibers there are these dark bands and the light bands. Okay, dark bands which are present over here, they are made up of a protein that is called as myosin. Light bands that are present, they are made up of actin. Okay, so myosin is a heavy filament, whereas the actin is a light filament. The diameter of the myosin is around 150 angstrom, and the actin filament's diameter is around 70 angstrom. This is a muscle fiber, and we can see there are these alternate bands, that is dark bands, light band, dark band, and the light band. Now what are these all labels that are given over here? That only we are going to study in detail. Okay, here this whole filament that we can see is the myosin protein. Okay, this blue colored filament that is present, it is a actin protein. So there is actin, there is myosin, again there is actin, and then there is a myosin protein. Now where we can see the actin, okay, this whole actin band where only actin filaments are present and no myosin filament is present, that is called as the I band. Okay, it is called as the I band. Whereas from this myosin filament, okay, this whole band is called as the A band. Okay, why they are called as I and why they are called as A band? I band is isotropic band, whereas A band is anisotropic band. Okay, so this I band only contains the actin filaments, and the A band contains only the myosin filaments. Now here, okay, we can see that there is a place where only the myosin filament is present and actin filament are not present, completely absent. So this zone is called as the H zone. It is labeled over here. Okay, so we have studied I band, only the actin filament region. We studied the A band where myosin is present, and we studied a region where the actin is completely absent, that is called as the H zone. And in the H zone there is a line of a protein molecule. That protein molecule is M creatine kinase, and the line is called as a M line. Okay, after this there is another line that is called as a Z line. Okay, this actin filament is connected to one another, okay, by a protein that is called as desmin. Okay, so जहाँ पे भी desmin present है, that line is called as a Z line. Here also there is one line, that is a Z line. So दो Z lines के बीच में का जो area है, जो region है, that is called as one sarcomere. It is labeled over here. It is called as a sarcomere, which is a functional unit of the muscle. And inside this sarcomere only, we will further study the sliding filament theory. How does a muscle contraction occurs, and for that muscle contraction this actin and myosin are going to play a very important role. So this is all about a sarcomere. Again, for a short revision purpose, we will again revise the things. Here there is actin filament, which is a thin filament, and then there is a myosin filament, which is a thick filament. Where the only actin filaments are present, that area or that band is called as the I band, which is a light band. Where the myosin is present, that area or that band is called as a A band. Okay, You can see the green label over here. I have already done it. And where there is no actin but only myosin is present, that zone is called as the H zone. In that H zone there is a M line which is made up of the M creatine kinase. There is also another line which is called as a Z line, and that Z line is present in the I band. The area or the region between the two Z lines is called one sarcomere, which is a functional unit. Okay, now let's see here. Okay, in this muscle fiber it is also given, this is the Z disc or the Z line, okay, which is present in the light region, that is the actin filament. This is the H zone where there is only the myosin that is present and actin is completely absent. Okay, this is one Z line, this is the another Z line, and in between this region the region which is present, that is called as the sarcomere. Okay, this is I band, A band, I band, and this is the M line that is given here. Here also in the black colored straight lines that we can see, that is the M line. Now when we talk about myosin, myosin is a protein molecule, and this protein molecule is made up of monomers, and that monomer is called as a myromyosin. Okay, myromyosin, these are the two heavy chains which are spirally coiled around one another. Okay, this is one heavy chain and this is the another heavy chain. This heavy chains, they have two heads. Okay, these two heads are globulin in nature, and these two heads which are basically made up of heavy myromyosin as well as the light myromyosin. The heavy myromyosin region has a site, or the head has a site which has ATPase activity, that is, it is going to hydrolyze the ATP and break the ATP into ADP and is going to release one inorganic phosphate. This one single head has two light chains and this head has two light chains. So there are total 4 light chains and two heavy chains in the head region. This region that we can see, it is a spirally coiled protein structure which is called as the tail. This part is the neck. Okay, that is the cross arm region, and the cross arm region and the head region, they are connected with a hinge, and because of the presence of a hinge, the head can move backward and front. Okay, so this backward and front movement of the head is very much important in the sliding filament theory. So this is about myosin, and myosin is made up of a single unit that is called as a myromyosin, which contains 2 heavy chains. Okay, that is 2 heavy myromyosin and 4 light myromyosin. Next one, this is the another protein that is present, and this is basically going to make the light band. Okay, so light band is made up of 3 proteins. One is the actin protein, the another is the tropomyosin, and the third one is the troponin. Actin is present in 2 forms. One is single unit and one is a polymer. Okay, monomer and the polymer. Monomer is called as the G-actin, but when many monomers come and bind together, they form a polymeric protein, this yellow color structure that we can see, and this is called as an F-actin. Okay, on this F-actin, the another protein, that is a tropomyosin, it wounds, and tropomyosin has 2 strands. Okay, and it is alpha helical strands which coil around the grooves of the F-actin. ओके, जहाँ पे भी F-actin के grooves होते हैं, grooves मतलब डिप्रेशन, वहाँ से जो ट्रोपोमायोसिन है वो बाउंड होता है F-actin के साथ. एंड ऑन द ट्रोपोमायोसिन के ऊपर कई जगह पे रेगुलर इंटरवल्स पे और एक थर्ड प्रोटीन प्रेजेंट होता है, दैट इज कॉल्ड एज द ट्रोपोनिन. Troponin is present at such a site that in a normal condition, in a relaxed condition, it is covering the area or it is masking the region of myosin binding site that is present on the actin filament. Actin filament के ऊपर, light filament के ऊपर एक site होता है जो myosin के head के साथ जाके bind होगा. Okay, this head or this site is not head, this site is covered by the troponin. Okay, during the time of muscle contraction, the troponin goes away because calcium comes and takes the troponin away, and these binding sites on the thin filaments, they get exposed, which can bind to the head region of the myosin. Okay, now let's go to the sliding filament theory. But before the sliding filament theory, okay, what happens that we have to see. This structure shows two things. One is, okay, this okay. But what happens before the sliding filament theory, that we have to understand. Okay, there is this axon membrane. We all know what is a neuron. Okay, neuron has the telodendrites, then it has the cyton body, axon, through which the nerve impulse is going to get transported, and finally it comes to the telodendrite region, and inside the telodendrites, the knob-like structures, there are these vesicles that are present. These vesicles, they contain a chemical transmitter which is called as the acetylcholine chemical transmitter is present. When the impulse comes towards this region, okay, this impulse activates these vesicles to go and fuse to the membrane of the knob-like structure, that is the telodendrite. When the fusion takes place, the acetylcholine from these vesicles is released into the synaptic cleft. Once they come into the synaptic cleft, okay, this is the motor end plate and a neuromuscular junction. Neuromuscular junction, where the neuron is in contact with the muscle. The muscle cell has some receptors that are present on it, which are going to receive the acetylcholine. Okay, and there is also an enzyme, that is the chlorine esterase enzyme, which is going to cleave or break the acetylcholine into acetate and choline. Some of the choline goes back into the vesicles again to produce more acetylcholine. What happens here? Okay, earlier when the muscle was in a resting state, okay, a potential difference was maintained. The sodium ions were present on the outside and the potassium ions were present on the inside. Okay, so on the outside it was more positive than the inside. We are talking about the sarcolemma. Okay, here is the sarcolemma. We are talking about the plasma membrane of the muscle when it was in a resting potential. But when this change takes place, that is when a signal or an impulse comes from the neuron and it activates the muscle fibers, all the sodium ions, they start traveling inside the sarcolemma by the process of active transport, and potassium ions, they start moving outside again by the active transport. This produces, or this creates, an action potential. What it creates? It creates an action potential, because this action potential is necessary to carry out the contraction of the muscles so that the muscles function and the body movement or the locomotion should take place. Okay, action potential. Because of this, the calcium ions, they start entering inside the T tubule, okay, that is present on the muscle fiber. From the T tubule, these calcium activate the sarcoplasmic reticulum, and the sarcoplasmic reticulum start releasing the calcium ions, because I have mentioned one point, that the sarcoplasmic reticulum is a reservoir of the calcium ions. Okay, these calcium ions, when they are released by the sarcoplasmic reticulum, what happens with the thin filament that we have to see now? Okay, here we studied that there is troponin. Troponin, you have to remember that it is made up of, again there are three types of troponins. Okay, there is troponin C, there is troponin I, and there is troponin T. Okay, so what this troponin C does is that the troponin C binds with the calcium. Okay, the troponin I, it inhibits the F-actin myosin interaction, and the troponin T binds to the tropomyosin as well as to the other components. कहने की बात ये है कि तीन टाइप्स के ट्रोपोनिन होते हैं. ट्रोपोनिन टी जो है वो ट्रोपोमायोसिन के साथ बाइंड होता है. ट्रोपोनिन आई जो है वो एफ एक्टिन को इनहिबिट करता है कि वो मायोसिन के साथ जाके बाइंड ना हो, और ट्रोपोनिन सी जो है वो वहीं पे रहता है जब तक कैल्शियम वहाँ पे नहीं आता. But as soon as the calcium comes, when the calcium is released from the sarcoplasmic reticulum, this troponin C has affinity for calcium, it binds with the calcium, and this whole troponin is removed from that region. Now when this troponin is removed, what is exposed? बाहर क्या निकलेगा? बाहर निकलेगा ऐसा साइड जो यहाँ पे दिख रहा है हम लोगों को. We had studied the head region of the myromyosin. Okay, and this is the cross arm, and this is the tail region. Okay, and this whole structure is a myosin filament. Okay, नाउ व्हेन दिस इज एक्सपोज्ड, इसके ऊपर हेड के ऊपर जो साइट है एटीपेज साइट, ओके, एटीपेज साइट हैज एंड देयर इज आल्सो अनदर साइट दैट इज द एक्टिन बाइंडिंग साइट। एक्टिन बाइंडिंग साइट जो है वो एक्टिन के साथ जाके बाइंड करेगी। एटीपी का वहां पे हाइड्रोलाइसिस होगा। एटीपी का जब हाइड्रोलाइसिस होता है तो एटीपी जो है वो एडीपी में कन्वर्ट हो जाता है और एक इनऑर्गेनिक फॉस्फेट को रिलीज करता है। दिस गिव्स एनर्जी। देन बिकॉज ऑफ दिस रिलीज्ड एनर्जी, ओके, व्हाट हैपेंस इज दैट दिस मायोसिन हेड इट स्टार्ट्स मूविंग इन an opposite direction, and this causes the sliding of this thin filament over the thick filament, okay, it slides, so that is why it is called as the sliding filament theory। इसका और एक डायग्राम है मेरे पास, वो अभी हम लोग स्टडी करते हैं, यहाँ पे दिया है, ओके, जो अभी वो तो सब बेसिक था, नाउ वी हैव टू स्टडी दिस, ओके। नाउ दिस इज द पोस्ट रिगर, दैट इज यहाँ पे जो है एक्टिन फिलामेंट इज इन रेस्टिंग पोजीशन, दैट इज स्केलेटल मसल जो है वो पूरा रेस्टिंग पोजीशन पे है। ये हेड है, ओके, मीरोमायोसिन का, ओके, यहाँ पे अभी कुछ भी नहीं हो रहा है, बट जैसे ही सिग्नल आता है, जैसे ही कैल्शियम आके ट्रोपोनिन को निकाल के चला जाता है, ट्रोपोनिन के ऊपर मायोसिन बाइंडिंग साइट होता है और मायोसिन हेड के मीरोमायोसिन के हेड के ऊपर एक्टिन बाइंडिंग साइट होता है, ओके। तो इन दोनों के यहाँ पे जो है बाइंडिंग होगा। When the binding has to take place, ATP is hydrolyzed into ATP and inorganic phosphate। तो यहाँ पे जो ये हेड है वो जाके बाइंड करेगा, किसको जाके बाइंड करेगा? एक्टिन मॉलिक्यूल को बाइंड करेगा, ओके। अगेन हियर यू कैन सी द थर्ड डायग्राम, इट इज बाइंडिंग एंड देन फाइनली इट इज मूविंग। यहाँ पे देखिए, यहाँ पे पावर स्ट्रोक होता है, यहाँ का एंगल अलग है और यहाँ का एंगल अलग है, तो ये पीछे की तरफ मूव हो गया, मतलब ऊपर का जो एक्टिन वाला जो फिलामेंट है वो उसके ऊपर स्लाइड हो गया, आगे की तरफ खींच लिया गया, ओके। यहाँ पे भी खींचा गया है एंड देन फाइनली द एनर्जी इज ओवर, नाउ नो मोर power stroke can carry out, and again this cross linking of the myromyosin and the actin has to be broken, so again the muscle comes into a relaxed state, again here this is the contraction and this is the relaxation। तो ऐसे होता है sliding filament theory। what happens in the sliding filament theory we will see over here, okay। let's start, this is the myromyosin head, okay। मीरोमायोसिन हेड जो है वो एक्टिन के साथ जाके क्या करेगा? बाइंड करेगा। यहाँ के भी जो मीरोमायोसिन के जो हेड है वो भी एक्टिन के साथ जाके क्या करेंगे? बाइंड करेंगे। व्हेन बाइंडिंग टेक प्लेस, पावर स्ट्रोक की वजह से ये वाला एक्टिन फिलामेंट यहाँ पे स्लाइड करेगा, ये वाला एक्टिन फिलामेंट यहाँ पे स्लाइड करेगा। तो स्लाइडिंग की वजह से एच जोन का जो लेंथ है वो क्या हो गया? ऑब्वियसली लेंथ कम हो गया, राइट। तो एच जोन का जो लेंथ है वो क्या हो गया? कम हो गया। आई बैंड जो है उसका भी लेंथ क्या हो जाएगा? आई बैंड का भी लेंथ जो है वो कम हो जाएगा। लेकिन लेकिन थिन फिलामेंट हो या फिर मायोसिन फिलामेंट हो, उनका लेंथ कम नहीं होगा। आई बैंड का लेंथ कम होगा बिकॉज ये जो है वो इस तरफ खींच लिया जा रहा है, ये भी इस तरफ खींच लिया जा रहा है, तो आई बैंड का जो लेंथ है वो कम हो जाएगा। एच जोन का जो भी लेंथ है वो क्या हो जाएगा? कम हो जाएगा। जो मायोसिन का फिलामेंट है वो कहीं पे भी स्लाइड नहीं करता। इमेजिन देयर इज अ सरफेस ऑफ वाटर एंड ऑन दैट देयर इज अ बोट, ओके। विद द हेल्प ऑफ अ रोवर यू आर मूविंग द बोट ऑन द वाटर सरफेस। तो बोट जो है वो मूव हो रही है लेकिन वाटर जो है वो वहीं पे है। सो ये जो मायोसिन है दैट इज देयर ओनली, उसका लेंथ कभी भी कम नहीं होगा। ओनली द लेंथ इज रिड्यूसिंग इज ऑफ द आई बैंड एंड द एच जोन। तो सार्कोमियर का भी लेंथ क्या हो गया? कम हो जाएगा ऑब्वियसली, बिकॉज दिस इज द एच जोन, सॉरी दिस इज द जेड लाइन एंड दिस इज द जेड लाइन। तो दोनों भी अगर फिलामेंट्स दैट इज अ थिन फिलामेंट्स एक दूसरे के पास नज़दीक आ रहे हैं तो जेड लाइन का भी जो लेंथ है वो क्या हो जाएगा? कम हो जाएगा। दैट इज सार्कोमियर का भी लेंथ कम हो जाएगा। अगेन ड्यूरिंग द टाइम ऑफ रिलैक्सेशन द सेम thin filaments go back on their original position and again the muscle cell undergoes relaxation process. So this is about the sliding filament theory, and this sliding filament theory was given by two groups of scientists. One group of scientists is A.F. Huxley and Ralph Niedergek in 1954, and the another group is H.E. Huxley and Jean Hansel. In the next video, we will discuss about the roles of biomolecules in the muscle contraction. Till then, keep on studying. Thank you very much.